All right, so let's get started on chapter 12, which is the central nervous system. All right, so I want to remind you of some definitions and terminology here before we go on, because this will continue to pop up, and especially on your tests as well. So in neurobiology, the term nuclei is talking about clusters of neurons in a particular area of the brain. So don't actually confuse that term with nuclei of cells. So just making sure you understand that. All right. So before I go into the slide, can anyone tell me the different parts of the central nervous system? What composes the central nervous system?
Brain, spinal cord.
Brain, spinal cord. Good. All right, so central nervous system, different from peripheral nervous system, includes the brain and the spinal cord. So this slide is nothing new. We've already talked about this already. Functions of the central nervous system. It has motor functions, sensory functions, and integrative functions. Which of those three is specific to the central nervous system versus peripheral? Integrative functions, right? All right, so again, motor functions include stimulation of a muscle cell contraction or a gland secretion. Motor functions are also a function of the peripheral nervous system. Again, nothing new there. Sensory functions, essentially detecting sensations within and without, outside the body, is also a function of the peripheral nervous system. And then integrative functions. This is an exclusive function of the central nervous system. Highlight that. This includes decision-making processes. So, for example, interpretation of sensory information, planning and monitoring movement, maintaining homeostasis, which is critical, and higher mental functions such as language, learning, cognition, motion, those kind of things. All right, so we're going to talk about the brain, then we'll talk about the spinal cord. And the same with this overall PowerPoint. We're going to talk about different portions of the brain, and we're going to talk anatomy of the spinal cord. All right, so the brain is anatomically continuous with the spinal cord and is directly or indirectly controlling most of the body's functions. Makes sense. So it's made mostly of nervous tissue, and it contains epithelial and connective tissues as well. It has internal cavities called ventricles that are filled with cerebrospinal fluid, which we'll talk about in just a little bit. And it is kind of like an energy hog, blood hog. So it receives about 20% of the total blood flow during periods of rest, and this reflects its requirements for huge amounts of oxygen, glucose, and nutrients. All right, so the spinal cord, again, two components here, brain and spinal cord. Spinal cord is a long tubular organ which is enclosed within a protective vertebral cavity. It blends with the inferior portion of the brainstem, something you probably know already, medulla oblongata. So the central canal is an internal cavity within the spinal cord that is continuous with the brain's ventricles. This is also filled with that same cerebrospinal fluid. Again, we'll talk about that in just a little bit. Highlight this slide. You must know this. The brain consists of 4 divisions. Each is distinct in the type of input it receives and where it sends its output. 4 divisions of the brain are as follows. The cerebrum, this portion kind of like in tan here. Cerebrum. I want to skip on down. Something that sounds very, very similar. Cerebellum. That's this kind of reddish portion. Don't confuse the two. Cerebrum here. Cerebellum. The diencephalon here in blue. And then the brainstem. All of which have various components and parts which we're going to talk about. General overview of the function is as follows. The cerebrum performs higher mental functioning, language, motion, different kinds of things, cognition. It interprets sensory stimuli, and it plans and initiates movement. The diencephalon here processes, integrates, and relays information. It maintains homeostasis and will regulate certain biological rhythms. The cerebellum is going to mostly be responsible for monitoring and coordinating movement. And the brainstem controls homeostasis, controls certain reflexes, monitors movement, and integrates and relays information. The most important portions out of all of these, kind of like the summary, I'm going to point out real quick. So the cerebrum is going to perform higher mental functions. Kind of the one you kind of think about. Let's start with that. When you think of cerebrum.
I went over my head.
Pretty good. Now you're good. All right, so the diencephalon, mainly going to relay information. It's like the shipping center in the brain, like telling everywhere to go except for certain particular. We'll talk about that. All right, so the cerebellum is going to monitor and coordinate movement. It's making sure that the movement is fluid, and it's involved in learning like muscle memory. It's involved in that muscle memory kind of portion. All right, so brainstem is going to maintain homeostasis. That's generally what you think of when you think of brainstem. It's going to be like your basal, like your heart rate, and respiration rate, and stuff like that. All right, so let's talk the basic structure of the brain and the spinal cord. So communication between gray and white matter connects different regions of the brain and the spinal cord with one another. So myelinated axons enable near instantaneous communication between locations. However, you do need to know the organization of the gray and white matter in the brain and the spinal cord is reversed. The spinal white matter is superficial when compared to the brain where it is deep. Also remember, white matter is myelinated and gray matter is unmyelinated. Okay, and here's a visual representation of what I've said. Basically, the white and gray matter are reversed in the brain versus the spinal cord. So white matter here on the inside versus on the spinal cord, it's on the outside. And then gray matter here, cortex, essentially, versus gray matter here. All right. So the next little portion talks about nervous system development. I don't really talk about that much. Essentially, with development here, you have the neural tube, which is split into the forebrain, midbrain, and hindbrain, which develops eventually into our structures in our mature brain. If you want to know more about that, there's lots of videos. I try not to talk too much about that because that gets really intense. You can take like a whole class over just that. All right, so let's talk for just a second about the brain. All right, we're going to talk specifically about all those different core divisions. We're going to talk sections and we're going to talk subdivisions over those. All right, so the cerebrum is the structure responsible for higher mental functions. Nothing new, we just talked about that. So it does have kind of distinct anatomical features, which you will need to know both here and in lab. All right, so sulci are shallow grooves on the surface of the cerebrum, versus gyri or gyrus, singular versus plural, are elevated ridges found between the sulci. Together, they're going to increase the surface area of the brain, which maximizes limited space within the confines of the skull. So it basically allows for more area with less room. Okay. There's another one that they don't really show you on here. It's kind of similar to sulcus. It would be a fissure. So if this is a hill and this is a valley, if gyrus are hills, the sulcus are valleys, you would have a really deep valley, which is essentially a fissure. That's another term you might hear. It's just a deeper form of sulcus, just so y'all know. I don't know why they didn't put that on there. All right, so again, we're still talking about the cerebrum here. The cerebrum. Let me go back real quick to show you the cerebrum, this tan portion here. We're going to be talking about subdivisions, specifically lobes. So remember when I talked about in our skeletal system chapter, we had the frontal lobe, parietal lobe, frontal bone, parietal lobe, bone, good Lord, I can't say. All of those different bones will correspond to these different lobes. So if you learn them then, you kind of already know them now. All right, so you have 5 lobes that are found in each hemisphere, both right and left sides of the brain. All right, so the frontal lobe is the most anterior of the lobes. So neurons in these lobes are responsible for planning and executing movement and complex mental functions such as behavior, conscience, and personality. That's why when people battle autonomy, personalities are kind of changed. Be kind of like zombies, have no personality. Because they're essentially scrambling up this frontal lobe, which is another interesting topic in history. If you ever want to look that up, you should definitely do it. All right, so the parietal lobe is just posterior to the frontal lobes. Neurons in these lobes are responsible for processing and integrating the sensory information and function in attention. The temporal lobe forms the lateral surfaces of each cerebral hemisphere. Neurons in these lobes are involved in hearing, language, memory, and emotion, which we're going to talk about some specific portions associated with language in just a little bit. I have a couple of videos on those. All right, occipital lobes are going to make up the posterior aspect of each cerebral hemisphere. Again, that's on this posterior side here. And neurons in these lobes are going to process all information related to vision. I always thought that was interesting because it's completely opposite of what the eyes are. So very back side, very farthest away. That always helped me to remember that. You also have insulas, which are deep when compared. They're underneath the lateral fissures on each side. And neurons in these lobes are currently thought to be involved in functions related to taste and functions involving viscera, which are basically organs. All right, so again, we talked about gray and white matter. Let's talk about the cerebral cortex. Very, very important. So this would be your cerebral cortex here. It's the outermost layer of the brain, which contains the gray matter. It's responsible for many higher order functions like language and information processing. That's where a lot of that integration is going to take place. All right, so the cerebral cortex is functionally the most complex part of the cortex. It covers underlying cerebral hemispheres, and all neurons in this cortex are what you call interneurons. So most of the cerebral cortex is what you call neocortex. It's so named because it's the newest evolutionary portion of our brain when compared to the allocortex, or the area connecting those in between them, called the mesocortex. The allocortex and the neocortex are basically, they try to make subdivisions of everything, and the neurons are connected just a little bit different in those areas, the neocortex versus allocortex. They're just basically parts of the cerebral cortex, but one is a little bit evolutionarily newer when compared. All right, so it has this huge surface area composed of 6 layers of neurons and neuroglia, whereas the allocortex only has 3. And the functions of the neocortex evolve around conscious processes, such as planning movement, interpreting incoming sensory information, and complex higher functions. So the neocortex is divided into 3 areas: the primary motor cortex, the primary sensory cortices, and the association areas. You need to know this. Motor cortices, cortex essentially. Primary sensory cortices, so motor, sensory, and association areas, which are integration centers. So nothing new here. It's just talking about 3 areas, and they're the same ones we learned previously. Motor, sensory, and integrated. All right, so the primary motor cortex is going to plan and execute movement. That's nothing new. We can tell that from its name. Primary sensory cortices, many sensory cortices, are the first regions to receive and process sensory input. Again, that makes sense. Look at the name. Right? Association areas, we're going to have a few different ones, but they're going to integrate different types of information. You can have 2 different ways. So unimodal areas integrate only one specific type of information, versus multimodal areas integrate information from multiple different sources, and they're going to carry out those higher mental functions. So again, integrative centers, just depending. Okay? All right, so this is everything just in a pictorial form. In lab, you'll have to learn some of these, not a whole lot. Today, in particular, we're going to be focusing on Broca's area here and Wernicke's area here, specifically on the left side of the brain. Okay? Those are going to be involved in processing and producing language speech. Right, this gives you an overview of every single area, what it does, type of cortex, location. I don't ask that you memorize that. That's way too much. I just ask that you know the general format. All right, so remember, we're still within the cerebrum here. We've not gone on to any other sections. We're still on the first of those 4 different areas of the brain. All right, so the basal nuclei, shown here, are found deep within each cerebral hemisphere, both left and right sides. So it's a cluster of neuronal cell bodies and it is involved with movement. So it has 3 different subdivisions. Generally, I just want you to know it's involved in movement. So it contains the caudate nuclei, the putamen, and the globus pallidus, which again, just parts of this basal nuclei, which is involved in movement. Okay, that's the important part to know. All right, so that was all gray matter. Let's talk white matter, still on the cerebral. So cerebral white matter can be classified as one of 3 types: commissural fibers, projection fibers, or association fibers. The commissural fibers are the ones we mostly talk about in here and in lab. We'll see some of these today, which if you're watching this later, you'll see some of this when we do dissections, specifically the corpus callosum. It basically connects the 2 areas of the brain. So commissural fibers are going to be connecting left and right. The biggest of this is the corpus callosum. There's some real interesting videos, some real interesting stuff. Happened way back in the 60s. They were trying to fix, basically this guy had horrible seizures, discommunication between the 2 hemispheres of the brain, and they cut his corpus callosum. Some of the seizures stopped, it kind of reduced quite a bit, but he could do different things with different eyes and like read something over here and do watch something over here. And then he could also do different things with different hands, like see something with this eye and draw it with this hand, versus if he tried to draw it with the other hand, he had no idea what it was. It's so totally awesome. I wish I could show it to you right now, but I think we're not going to have a whole incredible amount of time. If you want to see the video, I can find it for you. Just let me know, email me. But really cool stuff happens with these. So corpus callosum. All right, so projection fibers connect the cerebral cortex of one hemisphere with areas of the same hemisphere, other parts of the brain and spinal cord. And we'll see this in a picture in just a minute. The association fibers are restricted to a single hemisphere. They connect gray matter of the cortical gyri to one another. Okay, this is showing you exactly that. Okay, the only one that really connects the 2 hemispheres, right and left, are those commissural fibers shown here in blue. Okay. You also have projection fibers, which are actually these red portions here. Projection fibers. And then association fibers, which are shown here in green. Okay, so the limbic system is where we're going to go next. We're still in the cerebrum, but going on to the limbic system. All right, so this is found only within mammalian brains and is involved in memory, learning, emotion, and behavior. It includes several different parts: the limbic lobe, hippocampus, and the amygdala. So the limbic lobe is associated with the medial side of the cerebral hemisphere. It's associated with emotion, strong emotion. The hippocampus is in the temporal lobe and is connected to a prominent C-shaped ring of white matter, which is the fornix, which is also involved in memory and learning. The amygdala is anterior to the hippocampus. We'll see this in just a minute. It is also associated with strong emotion. In particular, it's associated with fear. Okay. All right, so just showing you the anatomy of that. Okay. So typically, when you think limbic system, you're thinking emotion. Usually what you associate with that. Okay. All right, so we're moving from the cerebrum to the diencephalon. We're moving from our first portion of the brain to our second portion of the brain. Okay, these are subdivisions, functions, all that kind of good stuff of the diencephalon. All right, so the diencephalon was that blue portion we saw earlier. It's at the physical center of the brain and is composed of 4 components: thalamus, hypothalamus, epithalamus, and the subthalamus. So each of these have their own nuclei that receive specific input and send specific output to other brain regions. So we're going to talk about each of these. The thalamus is the one you'll probably hear again, over and over again. The thalamus is the main entry route of sensory data into the cerebral cortex. Basically everything, all sensory information has to pass through there, with the exception of smell, has to pass through there to actually be integrated and understood. It has to pass through there in order to go anywhere. So it regulates cortical activity by controlling which input should continue on to that cerebral cortex. It basically goes from here out to specific areas in that cortex, wherever it may be. It's kind of like that UPS thing almost. All right, so the hypothalamus functions to include regulation of autonomic nervous system, sleep-wake cycle, thirst, hunger, and body temperature. It has several different parts too. The inferior hypothalamus is anatomically and functionally linked to the pituitary gland by an extension called the infundibulum. If they tell you in AMP2 that that's the only word you can use, technically it has another name, which is called the stalk. Much easier to remember. Infundibulum, stalk, if you take my AMP2, then you can use either term. It's basically the same thing. So hypothalamic tissue makes up the posterior portion of this endocrine gland, making up and releasing hormones. So the pituitary gland has the anterior and posterior pituitary. Basically what it's saying, the posterior pituitary is almost continuous with that hypothalamus, and essentially that's going to be making the hormones, the hypothalamus, not the posterior pituitary. Okay, the anterior pituitary is made of almost a completely different histologic tissue. One of them is a glandular kind of tissue versus the other is more of a nervous kind of derived tissue. And the difference of that, have you ever heard someone ask a question and say, oh, that's the way God made you? Embryologically, you are formed that way. That's why they're different. Different things are derived from different embryonic tissue. Okay. All right, so the point of that is just saying the inferior hypothalamus, that posterior pituitary is basically a continuation of that. All right, so mammillary bodies connect the hypothalamus with the limbic system. So remember that? We just heard about that. Limbic system is associated with what? Emotion. All right, so it receives input from the hippocampus and is involved in memory regulation and in behavior. All right, the epithalamus, which is, you would think with a name like epithalamus, it'd be like over it. It's actually kind of like almost in posterior superior to, almost it's kind of like, if this is your thalamus, it's kind of like behind and above almost. So the epithalamus is technically superior to the thalamus. It's kind of posterior as well. Most of its posterior superior bulk is an endocrine gland called the pineal gland. This secretes melatonin, which is a hormone involved in the sleep and wake cycle. So that basically regulates when you're waking up, when you're sleeping, and it does have a little bit on, some people say mood as well. All right, so the subthalamus, which is inferior to the thalamus, kind of almost anterior as well, is functionally connected with the basal nuclei. Again, we talked about in 3 different parts. Again, together they control movement. Okay. So movement is a complex feat. So there are lots of different sections that will coordinate and control movement. So it's not just going to be, oh, this one does this one does this. A lot of different sections control movement. And we'll see this again when we get to the cerebellum. All right, so this is just showing you anatomy of our diencephalon and the different parts of it. So again, thalamus here, epithalamus, and then posterior to that is the pineal gland, or inferior to that, I guess, is the pineal gland. The subthalamus here, mammary body. This is going to be your hypothalamus, and basically the inferior portion is going to run into this infundibulum and back into this posterior pituitary gland. The anterior pituitary gland is going to be a little bit different kind of tissue. But again, pituitary gland connected to the hypothalamus by the infundibulum or the stalk. Okay. All right, so what were our different parts we talked about before? We just talked about the diencephalon. What else did we talk about? Cerebrum. And then we're going to the cerebellum. 2 different things. Make sure to note that. The cerebrum and the cerebellum are not the same thing. All right, so the cerebellum is functionally connected with the cerebral cortex, the basal nuclei, the brainstem, and the spinal cord. Interactions between these regions together coordinate movement. So overall, the cerebellum is going to coordinate your movement. It makes so that your movements are nice and smooth and even, not jerky and jumpy. That's the part of the brain that's going to control that. So anatomically, it's divided into 2 cerebellar hemispheres. Again, right and left. and is connected by a structure called the vermis. The cerebellar cortex is the outer layer of gray matter in the cerebellum, similar to the cerebral cortex. It's the cerebellar cortex. Very similar, but again, different. All right, so the cortex is extremely folded, and this is your cerebellum here. This is going to be your brainstem. Okay, brainstem here and the cerebellum here. Important to know. All right, so the cortex is extremely folded and branching. It has white matter in it that's called the arbor vitae, and it's named that because it resembles tree branches. And we're going to see that in lab today, or if you're watching this later, we'll see it in lab whenever we do this. We may already see. All right, so white matter converges into three large tracts called cerebellar peduncles. 1, 2, and 3. 1, 2, and 3. This is the only connection between the cerebellum and the brainstem. All right, so that was the cerebellum. Now we're going to go on to our last section. We have 1, 2, 3. This is our fourth section, the brainstem. Lots of different divisions here, and we're going to talk about all of them. All right, so with the brainstem, its nuclei control many basic homeostatic functions. So heart rate, breathing rhythm, also reflexes are controlled here. So reflexes are our programmed automatic responses to different stimuli. So it functions in movement, sensation, and also maintaining alertness in addition to those homeostatic functions. Okay. Which it also contains several different things, but we're going to skip over that for now. All right, so this is just showing you connections. So this is a diencephalon, and then this would be the brainstem here underneath. So it has the midbrain, the pons, medulla oblongata, and then there's also reticular formation, which really isn't technically a part, but it is. I'll tell you about that in a little bit. All right, so let's talk about the different divisions here. Starting off with the midbrain, which is actually this superior portion of the brainstem. Again, this portion is the diencephalon, which is a totally different part of the brain. Just making sure. I've had a lot of students ask questions on that. All right, so the midbrain is inferior to the diencephalon, and it surrounds the cerebral aqueduct, which connects the third and fourth ventricles, the little spaces in the brain where that cerebrospinal fluid is going to flow, and we'll talk about that in a little bit. So it includes the following structures. So the superior and inferior colliculi, which are involved in visual and auditory functions. The substantia nigra, which works with the basal nuclei to control movement, which has some implication in Parkinson's. The red nucleus, which is involved in regulating movement. And the midbrain tegmentum, which contains components of the reticular formation. Again, it's almost like it's almost like the wires running through the center of all of this, the brain. So we'll talk more about that. All right, so the pons is inferior to the brainstem. It contains descending motor tracts coming from the brain through the spinal cord to their destination, usually carrying motor information. Descending motor tracts, some of which pass through the pons en route to the spinal cord. Pontine nuclei have many roles, including regulation of movement, breathing, reflexes, and complex functions associated with sleep and arousal. So again, several different sections that control different things, similar things. Again, it's very complex. Next, the medulla oblongata, which probably some of y'all remember from The Waterboy. That's like #1 movie of how people remember this. Medulla oblongata is the most inferior structure of the brainstem and is continuous with the spinal cord of the foramen magnum. So if we remember back to our bones, what's the foramen magnum? You remember? What does it look like? Can you describe it? It's like the big hole at the base of the skull. Yeah, good. So all these terms we've been learning throughout the semester are coming back to us. All right, so the medulla oblongata has several different parts. So it contains the pyramids, which are on the anterior surface of the medulla. They almost look like little projections almost. They're called pyramids because of the way they look. But basically they contain motor neuron fibers that travel from the cerebral cortex to the spinal cord. The right and left corticospinal fibers decussate or cross over within the pyramids. The motor fibers originating from the right side of the cerebral cortex descend through the left side of the spinal cord and vice versa. Also, the posterior columns, which are paired tracts of white matter that carry sensory information from the spinal cord, again going upwards, ascending. All right, so the posterior columns also decussate within medulla, so sensory information, again, crossover from the right side of the spinal cord is processed by the left side and vice versa. So you also have, there's several different names for this one as well, olive, literally, that's how you say it. You can also hear it called olivary nuclei. That's a little bit more common in some books versus this, it just depends. All right, so these are lateral to each pyramid, and it receives sensory fibers from the spinal cord and directs information to the cerebellum. You also have several cranial nerve and reticular formation nuclei found in the medulla, but again, we'll talk more about that here. So reticular formation is a collection of over 100 nuclei found in the central core of the 3 brainstem subdivisions, making this one of the most complex regions of the brain. So it's almost like you have you ever seen those Twizzlers that are like filled? It's like the filling portion, or maybe like a donut. It's the filling portion of the donut. Okay, that's the centermost portion. It's like wires running through the center of all 3 of those different parts in the brainstem. All right, so input is received from multiple sources, including the cerebral cortex, limbic system, and sensory stimuli, and the output is sent through the entire brain and the spinal cord. So the central nuclei, the center of the reticular formation, is going to function in sleep, pain transmission, and mood. Nuclei surrounding the central nuclei serve motor functions for both skeletal muscles and the autonomic nervous systems. Other nuclei are instrumental in homeostasis of breathing and blood pressure, and lateral nuclei play a role in sensation and alertness and in activity levels of the cerebral cortex. So basically what this is saying here is this is a super complex portion of the brain that does a lot of different stuff. Look at all of this. Functions in sleep, pain transmission, and mood, motor functions, breathing, blood pressure, homeostasis, sensation, alertness, activity levels, a lot of different stuff. A lot of those homeostatic norms are monitored by specifically this section. Just crazy. All that information. Okay. All right. So if you are just kind of blown by all of that information, these slides are really good. They give a summary. Okay. Kind of like a summary. All right. It talks about the 4 different divisions. Okay. There's 2 on this first one. And then it talks about the structures within that and the substructures within those and their functions. This is a super great slide. A lot of this gets really overwhelming. Use the broken down portions instead of trying to learn every single last word. Okay, that's what I recommend. Okay, so you have to know the 4 divisions. You have to know some of the subdivisions and the subdivisions of those and the functions overall. All right, that was the brain. Let's talk about protection of the brain. And then we're going to go on to the spinal cord. All right, so there's 3 features within the protective shell of the skull that provide shelter for the delicate brain tissue. And you feel, as we see this one, we are going to see this in lab. The brain is actually not quite as durable as we think it is. It almost has the consistency of like Jello almost. Or that's probably a little too firm for it. Well, you'll see in that. You'll see. All right, so how do we protect this brain? You have cranial meninges, cerebrospinal fluid, and the blood-brain barrier. So the cranial meninges are 3 layers of membranes that surround the brain, and also we're going to have spinal meningeal layers as well, which are basically the same thing. We'll have cerebrospinal fluid, which is a fluid that bathes the brain and fills the cavities, and the blood-brain barrier, which prevents many substances from entering the brain and its cells from the body, which again is no particular one anatomical location. It's really a function of those cells, essentially those astrocytes in the central nervous system and the endothelial cells basically a component of those two. All right, so the cranial meninges are composed of three protective membrane layers: the dura mater, the arachnoid mater, and the pia mater. In between those, you have other things, but really that's the important thing. So these are arranged from superficial to deep. Epidural space, the dura mater, which is our meningeal layer, the subdural space, the arachnoid mater, which is our meninge, the subarachnoid space, and the pia mater. Again, those are going from outside inwards. All right, so the epidural space is only really kind of a space for blood vessels. It's a potential space, as the dura is normally tightly bound to bone, only allowing for the passage of blood vessels. So basically, space is basically just blood vessels. All right, so the dura mater, or the dura, is the thickest and most durable of the three meningeal layers. It is a double-layered membrane. So you have the periosteal dura and the meningeal dura. The periosteal dura is the outer layer, is attached to the inner surface of the cranial bones, and it functions as a periosteum with extensive blood supply in the epidural space. The meningeal dura is the inner layer, and basically it's avascular, so any of that nutrients and stuff have to be diffused through. It's mostly fused, creating a single inelastic membrane, except in regions where cavities called dural sinuses are found. Those dural sinuses are going to come into play when we get to AP2. You'll have to learn a bunch of them, have to learn names, have to learn functions. We'll talk more about that when you get to AP2. Basically, dural sinuses are venous channels, basically veins. And they're going to drain specifically, in addition to blood, also cerebrospinal fluid. And again, when you're talking about a vein, well, when you're talking about a vein, generally, you're talking about deoxygenated blood. It's not always the case because that's not technically the actual definition of a vein. It's actually going back towards the heart. That's your technical definition, because when you get to the heart, veins versus arteries, the pulmonary, that's all kind of different. But technically, traditionally, what you think of with veins is deoxygenated blood. And that's what you see here. All right, so the subdural space is a serous fluid-filled space, and it houses veins that drain the blood from the brain. The arachnoid mater is another one of our meningeal layers, which is the the subdural space. It is so called arachnoid mater because of its thin web-like appearance. So the arachnoid trabeculae anchor the arachnoid to the deep pia mater, those two meningeal layers. The arachnoid granulations, or villi, project superficially through the meningeal dura into the dural sinuses, and they are going to allow for the return of cerebrospinal fluid into the bloodstream, which is something that we want because we have to replace that blood. We'll see that in just a minute. The subarachnoid space contains the major blood vessels of the brain and is filled with cerebrospinal fluid. Our last meningeal layer is the pia mater. It's the only meningeal layer in physical contact with the brain tissue. So it follows every contour of the brain, covering every sulcus and every fissure. So they put fissure here, but they couldn't put it in the last one. Sulcus versus fissure, again, one is more shallow versus one is deeper. Shallow versus deep. So it is permeable to substances in the brain in the extracellular fluid and cerebrospinal fluid, and it also allows for substances to move between these two fluid compartments, extracellular fluid and cerebrospinal fluid. So this helps to balance the concentration of different solutes found in each fluid. Again, simply by diffusion. Okay, and this is just showing you a pictorial representation of what I just said. Meningeal layers, brain protection, same thing, only zoomed in. All right, so those were our meningeal layers. Let's talk about our ventricles. So 4 ventricles within the brain are linked cavities that are continuous with the central canal of the spinal cord. They're lined with ependymal cells. Ependymal cells, again, are neuroglial cells that are specific to the central nervous system. Coming back to haunt us now. So these are glial cells with a similar look to ciliated simple columnar epithelial tissue. They're filled with cerebrospinal fluid. These ventricles are filled with cerebrospinal fluid. All right, so let's talk about these 4 ventricles here. You have the left and right lateral ventricles, or if you want to call them first and second ventricles, that's the same thing, it's fine. Left and right lateral ventricles are found within their respective cerebral hemisphere, left or right. Third ventricle is a narrow cavity found between the two lobes of the diencephalon. It is connected to the lateral ventricles by the ventricular foramen. Again, foramen is basically a little hole. All right, so the fourth ventricle is between the pons and the cerebellum. It's connected to the third ventricle by the cerebral aqueduct, which will come into play here in a minute. It's basically a small passageway through the midbrain. It's continuous with the central canal of the spinal cord, and it contains several posterior openings that allow cerebrospinal fluid in the ventricles to flow into the subarachnoid space, which will be important here in just a minute. Okay, so these are your ventricles here. They're going to have cerebrospinal fluid. They're going to protect the brain. They're going to keep the brain essentially functioning normally. And again, it isn't mentioned here, but the cerebrospinal fluid does also flow into the spinal cord, which we'll talk about. Right. Choroid plexuses are where the majority of the cerebrospinal fluid is manufactured. This is found in each of the four ventricles where blood vessels come into direct contact with ependymal cells. And remember, those were the glial cells that are going to be involved in circulating and, to some extent, manufacturing this cerebrospinal fluid. So we talked a lot about the cerebrospinal fluid. Let's talk about what it actually is. So cerebrospinal fluid, or CSF, is clear, colorless liquid, which is similar in composition to blood plasma, which honestly, blood plasma, a lot of it is water. Okay? So it protects the brain in the following ways. It cushions the brain and maintains that constant temperature within the cranial cavity. It also removes waste and increases the buoyancy of the brain. This keeps the brain from collapsing under its own weight. Okay. So now we've talked about all this. What about circulation and what about producing? So cerebrospinal fluid production and removal occurs constantly. So every 5 to 6 hours, you have a completely new cerebrospinal fluid. Isn't that cool? Isn't that insane? Every 5 to 6 hours. Dang. All right, so basically what happens here, we have a little picture on the next slide. Fluid and electrolytes will leak out of capillaries of the choroid plexuses and into the extracellular fluid of the ventricles. Basically, our capillaries have these specific type of connections between them that allows for this to happen. They are, out of the 3 types of capillaries, they are sinusoidal, they're leaky, so to speak, when compared to other vessels capillaries are. Okay. So once this happens, it's taken up into the ependymal cells, which are right next to it. You're going to see in just a minute. And then that's going to be secreted into the ventricles, changed up a little bit by those ependymal cells, changed into cerebrospinal fluid. This is circulated through and around the brain and the spinal cord. Again, those are connected, but in the subarachnoid space, and is assisted by the movement of the ependymal cell cilia. Again, those little finger-like structures, those little hair-like structures on the top that continually beat and circulate that fluid. So some cerebrospinal fluid is reabsorbed into the venous blood in the dural sinuses via the arachnoid granulations. That's how this whole process happens over and over again. Some of it's taken up and some of it is continually put out. Okay, this is the same thing here. It's just in the pictorial form. So I'm just going to go real quick through this because I think it was a little bit wordy there and it might help you all to look at some of these pictures. All right, so fluid and electrolytes will leak out of the capillaries here. And look what's right next to it. Ependymal cells. They're going to leak out here and essentially go into these ependymal cells. And that's essentially going to change it around just a little bit. And it's going to secrete that into basically the choroid plexus, which is like, basically we're going to make our cerebrospinal fluid here. Okay, it's going to be secreted out. We have all these nice little cilia here. So ependymal cells secrete extra cerebrospinal fluid into the ventricles. Okay, and that's going to circulate all around here. So cerebrospinal fluid circulates in and around the brain, and the spinal cord, and the subarachnoid space. Okay. So some of that cerebrospinal fluid, and that's showing these little granulations here, some of that cerebrospinal fluid is reabsorbed into the blood in the dural sinuses via these arachnoid granulations. So it's continually happening over and over again. Make some and take some, make some and take some. So about every 5 hours or so, you have completely new cerebrospinal fluid. All right, let's talk about this one real quick. The blood-brain barrier. I know we've already kind of talked about it, but we'll talk about it just a little bit more. All right, so the blood-brain barrier is a protective safeguard that separates the cerebrospinal fluid and the brain extracellular fluid from chemicals and disease-causing organisms sometimes found in the blood plasma. Hopefully not. Hopefully you don't have an infection that's systemic and it's just going everywhere and traveling through the bloodstream and craziness, but it could happen. So it mainly consists of simple squamous epithelial cells, or in particular endothelial cells of the blood capillaries. So all that is, endothelial cells are lining the capillaries. It's essentially like a single layer of cells, but what they are is simple squamous epithelial cells. So when we're talking about simple squamous epithelial cells, again, one layer, that's how you get that simple. Squamous meaning what? Flattened, scale-like cells. So really all this is saying, endothelial cells, they have a certain name because they're in a certain place, but they're the same thing. It's one layer of these very thin, kind of flattened cells, okay? So again, it consists mainly of those endothelial cells, the capillaries, their basal lamina, and the astrocytes. Again, those astrocytes have these little projections which will wrap around those capillaries, and again, functions of those two together, those little feet around there, and the endothelial cells will contribute to the blood-brain barrier. That's really what it is. So neighboring endothelial cells are bound together by many tight junctions, which prevents fluids and molecules from passing between them. It depends on a certain area. That's really subjective. But it does have a limited capacity for movement of molecules and substances into and out of the cell by endocytosis and exocytosis. So again, two things contribute to the blood-brain barrier: those endothelial cells and those astrocytes that feed on those, which wrap around the capillaries. All right, so again, there's no one specific, like, oh, I'm going to point at that. Oh, that's the blood-brain barrier. No specific one little place where this is. It's found throughout the entire brain. That's the purpose of this slide here. It's just talking to you about that. I'm going to go ahead and skip over this. Again, this really summarizes it. The blood-brain barrier is found throughout the entire brain and is actually a property of the capillaries and actually those astrocytes rather than a distinct physical barrier. Okay, and this is shown here. This is your typical capillary versus in the brain, they're a little less leaky. And you also have extra support here in those astrocytes, those glial cells, those supporting cells in green here. All right, so we're going to talk real briefly about infectious meningitis. So this is potentially a life-threatening infection of the meninges in the subarachnoid space. So inflammation is going to occur, causing classic signs of headache, lethargy, stiff neck, and fever. So essentially, to diagnose this, you have to examine the cerebrospinal fluid for infectious agents. I say infectious agents because there's many different kinds, and your white blood cells. So different kinds of white blood cells can indicate different things. For example, if it's a bacterial infection, you might have elevated levels of neutrophils, which are a certain kind of white blood cell versus another. So bacteria and viruses are the most common cause of agents of infectious meningitis, but you can also have fungal as well. There's not very many real bad fungal infections in humans as far as like compared to bacterial or viral, but it does happen. All right, so viral infectious meningitis is generally mild and resolves within 1 to 2 weeks. Bacterial infectious meningitis is the bad ones we think of. Can rapidly progress to the brain, and it essentially a lot of times involves death. You have to have an aggressive antibiotic treatment, and some common forms are preventable with vaccines. Fungal is very interesting here. It's pretty rare, but it's usually only found in immunocompromised people. So somebody who is taking an immunosuppressant, somebody who has an immune system that doesn't work properly, either from genetics or whatever it might be. Maybe they have something like, AIDS, cancer, anything like that. They're susceptible to these opportunistic pathogens, which they would not be susceptible to otherwise. Basically, our opportunistic pathogens, you'll see this in micro, they see an opportunity and they take it. Your immune system doesn't work properly, or if we can't defend against this, well, I would normally be a good little bacteria on your skin, but now I'm going to cause a real bad infection because you can't keep me in check. Something like that, which meningitis is a little bit different. All right, so let's talk about, we talked about the brain and protection for the brain. Now let's talk about the spinal cord real quick. I focused less on the spinal cord, a little bit more on the brain, generally with the tests on these. All right, so the spinal cord is composed primarily of nervous tissue. Thank you for that. Who would have known? It's also responsible for both relaying and processing information. This serves two primary roles. It serves as a relay station and a processing station. All right, so the brain's meninges pass through the foramen magnum, that hole at the bottom of the skull, has a purpose, foramen magnum, to provide continuous protective covering of the spinal cord and distal nerves at the base. There's three spinal meninges, which include the dura mater, the arachnoid mater, and the pia mater, which is structurally similar to the brain meningeal layers. Exception is that the spinal cord dura has only one layer and the pia mater has some structural enhancements, which we don't really have to go through right now at this very moment. And this is just showing you the spinal cord and its meningeal layers here. Okay. All right, so spinal nerves we talk about in chapter 13. We'll talk a whole lot about our spinal nerves. We'll also talk about cranial nerves, which you'll have to learn all 12. Have to learn the spinal nerves, all kinds of good stuff. All right, so spinal nerves are components of the peripheral nervous system. So going out from that central nervous system to all of our basically nerves. These are nerves, these are spinal nerves, the peripheral nervous system. So they're going to carry sensory and motor impulses depending on the nerve to and from the spinal cord. So projections are visible on each side of the spinal cord between the vertebrae and are called posterior and anterior nerve roots. So roots of the spinal nerves extend inferiorly from the conus medullaris and fill the remainder of the vertebral cavity. This bundle of spinal nerve roots is called the cauda equina due to its horsetail-like appearance. So basically, your spinal cord technically stops at the conus medullaris, and you have these little, like it literally looks like a little, like a horsetail, like a little waterfall underneath that of nerves, which is called the cauda equina. So again, the conus medullaris is that cone-shaped termination of the spinal cord. The cauda equina is those projections of nerves coming off of that. Okay. I would highlight conus medullaris and highlight the cauda equina. I would highlight those two. So conus medullaris here, the cone-shaped termination of the spinal cord, the cone-shaped termination of the spinal cord, and the cauda equina, that kind of horsetail appearance of nerves coming off that, popping off the conus medullaris. Okay. All right, so this is our spinal cord anatomy. I'm going to run through this real quick because we can talk more about this in lab. All right, so you have a central canal with your spinal cord. It's filled with cerebrospinal fluid and is seen in the center of the spinal cord. It's surrounded by two thin strips of gray matter or the gray tissue. The spinal gray matter makes up three distinct regions found within the spinal cord: the anterior horn, posterior horn, and the lateral horn. So named because of the location. All right, so the anterior horn or ventral horn gives rise to the anterior motor nerve roots that are involved in somatic motor functions. So skeletal muscle contractions, the name, we understand. Skeletal muscle contraction, there are motor nerve roots. Makes sense. The posterior horn or dorsal horn gives rise to the posterior sensory nerve roots, which are involved in processing incoming somatic and visceral sensory information. Makes sense. Sensory and sensory. The lateral horn is found only in the spinal cord between the first thoracic vertebra and the lumbar region. It contains cell bodies of neurons involved in the control of viscera via the autonomic nervous system. Autonomic, you're thinking fight or flight, those automatic functions. Okay, this is our overview here. Okay, So with white matter, you have what's called ascending and descending tracks, going towards, a lot of times towards the brain, and a lot of times away from the brain. So it contains axons of neurons that travel to and from the brain, allowing the spinal cord to function as a relay station. Ascending tracks carry various kinds of sensory information, again, going towards the brain. Sensory information, we have to take what we see, integrate it, and then put it into the next section, descending tracks, which transmit motor information. Okay. A lot of this, if you just break it down into those three different functions, sensory, motor, integrative, it really makes a lot of sense. Okay. All right, so descending tracks again transmit motor information from the brain down the spinal cord to whatever gland or muscle they need to go to. Okay, this next section is talking about the role of the central nervous system in sensation, and then we talk about motor functions as well. All right, sensory stimuli are those effects that cause the senses to respond. So multiple sensory stimuli from different regions of the brain can be pulled together into a single mental picture. Maybe like you're really hungry and you see this beautiful looking piece of cake. It smells so good. You can just taste it right now. Pulling all kinds of different sensory information. And it's kind of making me have questions about that. All right, so each of these separate stimuli reaches the brain in the following two-part process. The stimulus is detected by the neurons in the peripheral nervous system, which special senses is what I was talking about more, but that's okay. And is sent as sensory input into the central nervous system. And the central nervous system, the sensory input is sent to the cerebral cortex, again, that outside portion, going outwards from the thalamus, which smell is different, it doesn't go through the thalamus, but to that outside portion, that gray matter, little folds, sent to the cerebral cortex for interpretation. When the central nervous system has received all different sensory inputs, it integrates them into a single perception, a conscious awareness of sensation. So sensations can be grouped into two basic types, which again, I specified. I was talking about special senses, but we didn't get to there yet. So special senses versus general senses. Our next, actually, no, not next chapter, chapter 15 is talking all about special senses. So vision, hearing, equilibrium, smell, taste, sensations. Versus general senses are detected by sensory neurons in the skin, muscles, or viscera. So they can be general somatic senses, so touch, stretch, joint positioning, pain, temperature, versus general visceral senses like your internal organs. All right. So this one, we do talk about some first, second versus third order neuron. I might ask you maybe one question over this. So first order neurons will detect initial stimuli in the peripheral nervous system. The axon of this neuron then synapses on a second order neuron. That's the first one really making contact with peripheral nervous system. So the second order neurons are usually interneurons, which are located in the posterior horn of the spinal cord in the brainstem. It relays the stimulus to the third order neuron. So this one to here, and then to our third order neuron. So the third order neuron is an interneuron, which is found in the thalamus. This will deliver the impulse to the cerebral cortex, again with the exclusion of smell. All right, so the thalamus relays most incoming information to the primary somatosensory cortex. So again, a specific part of that cerebral cortex in the postcentral gyrus. It's talking about a specific area, basically. So each part of the body is represented by a specific region of S1, or the somatosensory cortex, and a type of organization called somatotopy. So mapping of the primary somatosensory cortex illustrates that different parts of the body are equally represented. So more S1 space is dedicated to, for example, the hands and the face, which represents the importance of manual dexterity, facial expression, and speech to our existence. So the unequal representation of human body parts in S1 is exemplified by a horrible thing, a horrible, horrible picture. I hate whoever came up with this. But it's represented by what you call a sensory homunculus. I'll show you this on the next slide. Yeah, you can learn. This is the visual representation of that. Basically, what it's saying here, this is our cortex here, our somatosensory cortex. More area is given to things like the fingers, the hands, the lips, and that's what they're trying to illustrate to you here. But why couldn't you just give me this? I'd rather have this versus this monster thing here. You'll, if you really like this one, just wait. You're going to get another one. This is the sensory homunculus. What do you think the other one's going to be? Motor, like movement? Yeah. Yeah, just wait a minute. You'll see. All right. So let's talk about voluntary movement here. All right. So planning and coordination of voluntary movement is carried out within the central nervous system. We've already talked about that. So it involves motor areas of the cerebral cortex, the basal nuclei, the cerebellum, and the spinal cord, basically everywhere involved with movement. So 3 types of neurons are directly involved in eliciting and muscle contraction. Upper motor neurons with cell bodies in the motor area of the cerebral cortex, or some in the brainstem. Axons descend through the cerebral white matter to the brainstem and the spinal cord, and they synapse with local interneurons. Local interneurons will pass messages from upper motor neurons to the neighboring lower motor neurons. And then the cell bodies of the lower motor neurons reside in the anterior horn of the spinal gray matter, and axons, components of the peripheral nervous system, exit the central nervous system to innervate the skeletal muscles. Basically, your brain is telling your muscle to move. Again, nothing crazier than what we've already seen. Okay. All right, so the upper motor neurons are also located in certain nuclei of the brainstem and work to maintain posture, balance, and body position, especially during locomotion, during walking, whatever you want to talk about. Also, they produce motor responses to sensory stimuli. A map of the upper motor neurons and the primary motor cortex and motor homunculus resemble a sensory map and homunculus. So basically, there's disproportionately more cortical area devoted to like lips, tongue, and hands, which signals are the importance of vocalization and manual dexterity to human survival. Here's your other one. So again, more areas devoted to different things, and that's what they're trying to illustrate here. I think this one's worse because he's red. He looks kind of violent over here. But yeah, I would not ask you to do anything with this dude here on a test. Really just know that more area is devoted to certain things. Okay. All right. So we already talked about the cerebellum. Basically, the gist of this slide is saying that the cerebellum is involved in motor learning and is involved in like smooth movements with your muscles. So really, if you didn't have a cerebellum, all your movements would be jerky. They'd be really uneven and just jerky. That's really the function of that slide there. All right, this is talking about central nervous system and maintenance of homeostasis. All right, so if I could highlight any slide for the rest of the semester, like that was kind of summarizing anything we've talked about. This one, we've talked about this so much. I keep coming back to this over and over again. What 2 systems are going to regulate everything? What do you think? Nervous system and endocrine system. I beat you over the head with that. This is the overview slide of that. Okay? Nervous system and endocrine system are the main systems dedicated to maintaining homeostasis. They work together, but each has its own mechanisms for performing that vital homeostatic maintenance. So the endocrine system is going to secrete hormones. Okay? They secrete hormones directly into the blood. They don't act on nearby cells, they secrete it directly into the blood. And it regulates functions of other cells, target cells. So the nervous system sends action potentials. They're going to either excite or inhibit target cells, which are usually very close by. The nervous system usually has farther reaching effects and generally speaking, more long-term effects a lot of Right, so again, actions of the endocrine system are typically slow, but they are generally longer acting versus nervous system are immediately almost acting, but they tend to have a short time. Right, so two structures of the central nervous system are concerned directly with maintaining homeostasis: reticular formation and the hypothalamus. So reticular formation controls functions of many different internal organs as well as aspects of behavior. So reticular formation, that was a portion of our I've lost my train of thought. Brainstem. There we go. It's a portion of our brainstem. Remember that. Yeah, I've done too much talking. We go all the way back to reticular formation is a portion of our brainstem. So remember, this is going to be controlling like our respiration, our heart rate, those vital kind of autonomic functions. So the hypothalamus, among other things, will regulate body temperature and feeding. Darn, I hate my hypothalamus. So reticular formation and the hypothalamus have many different interconnections that enable them to coordinate many different homeostatic functions. All right, so again, maintenance of vital functions like heart pumping, blood pressure, and digestion are largely controlled by the autonomic nervous system to an extent, and again, it regulates the function of the body's internal organs. Although the autonomic nervous system is a component of the peripheral nervous system, it is controlled by components of the central nervous system, mainly our hypothalamus. All right, this next section is talking about higher mental functions, so language, cognition, things like that, which I actually took a whole class over cognition, so you can talk all kinds of stuff on cognition, but right now we're going to kind of gloss through this a little bit. All right, so cognition, you need to know the definition. It's a collective term for a diverse group of tasks performed by the association areas of the cerebral cortex. I'm going to kind of skip over that a little bit. Okay. This is the one I really wanted to focus on, language. So language refers to the ability to comprehend and produce words through speaking, writing, and or signaling, and to assign and recognize symbolic meaning of a word correctly. You will need to know these probably, if not for your exam 3, definitely for your final exam. I would know this. So Broca's area is in the frontal lobe, left side only. It is responsible for production of language. So it includes planning and ordering of words with proper grammar and syntax. Wernicke's area is in the temporal lobe, and it's on the left side only, and is responsible for understanding language and linking a word with its correct symbolic meaning. Aphasia is a language deficit. So you can have Broca's aphasia, Wernicke's aphasia, lots of different kinds of aphasias. So it occurs when either one of these two critical areas is damaged. A lot of times that damage happens with a stroke in that particular area. So when Broca's aphasia, you have an inability to correctly plan and order grammar and syntax. So the words and phrases are oftentimes repeated. Grammar, syntax, and the structure of individual words are in disorder. And it can be frustrating for patients because their language comprehension is intact. They can understand what they want to say, they can understand what you're saying to them, and they can understand that they're not saying the right thing, but they just can't do it. Their cognition is not impaired, it's just they can't do it. It's all kinds of trouble. Versus Wernicke's aphasia, they have an inability to understand the language. They can, let me put the word can in there, they can speak fluently with adequate grammar and syntax, but the words and meanings are not correctly linked. So they may say something, but they don't understand what they're saying. They cannot realize what they're saying makes no sense, and language comprehension is not intact. So I wanted to show you just real quick, these couple of videos. First, we'll start off here with Broca's aphasia. It might be kind of low, so trying to hope for the best here. Trying to hold high. This in particular is Broca's aphasia. This is somebody, they're interviewing somebody with Broca's aphasia. You might have to listen real close.
Can you tell us your name?
Hi,
Mike Caputo.
And Mike, when was your stroke?
I was 7 years ago,
Okay.
and
And what did you used to do?
well, worked Autodesk, 7 sales and worldwide, and very good. Yeah,
Okay. And who are you looking at over there? When you turn your
that's
head.
my wife.
Okay. And why is she helping you to talk?
She's a speech.
So you have trouble with your speech.
Yeah,
And what's that called?
aphasia.
All right. And so why don't you work now?
I do.
And what do you do now?
Voices of Hope aphasia.
And what is Voices of Hope?
Peterburg, and Dr. Hinckley, and myself, founder for me, and members, the members, probably 6 people.
So 60 people are part of Voices of Hope,
Yes.
which is an aphasia support group
Yes.
that you founded.
Yes.
And Dr. Jackie Hinckley is part of that.
Yes.
So pretty cool. That was Broca's aphasia. This is a patient who's doing pretty good with Broca's aphasia. Imagine having to live your life like that. He can understand anyone who's trying to talk to him and see what he wants to say, but he just can't. So pretty interesting. So with that one, the cognition is still there. Broca's aphasia. This is Wernicke's aphasia. He's very happy though.
Hi Byron, how are you?
I'm happy. You're pretty. You look good.
What are you doing today?
We stayed with the water over here at the moment. Talk to the people over them over there. They're guiding for them at the moment. They'll save in the moment. He'll water first for him. Good luck for him.
And we're on a cruise and we're going to
We
take
will
a
see
tour.
right here, and they'll save their hands right there. Yeah.
And what were we just doing with the iPad?
Right at the moment, they don't shut darn thing.
The iPad that we were doing. Like here?
I changed for me and changed hands for me. I was happy. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them. This girl was fairly good and happy. And I play golf.
He always kind of makes me smile. He's so very happy. He's saying happy over and over again. All right, what did y'all think about those two? It was real interesting. So with this one, the actual understanding of what he's saying, he has no understanding of what he's saying. So, but at least he's happy about it. All right. So what did y'all think of those? Any comments?
That Broca, the way he speaks is kind of broken, and so kind of makes it a little more relatable in my mind.
Yeah. Sometimes I talk like that just normally, and I'm not, I don't have any aphasions. My Broca's very struggling sometimes. All right, let's finish this up real quick. We don't have too much more here, but like maybe 6 slides or something. All right, so there's 2 basic types of memory, declarative and non-declarative. So declarative is like your factual memory, and non-declarative is like your, it's not your muscle memory, but it's like your procedural memory, your skill memory. All right, so declarative and non-declarative memory can be classified by length and time in which they're stored. Immediate memory is stored only for a few seconds and is critical for carrying out normal conversation, reading, and daily tasks. Short-term or working memory is stored for several minutes and allows you to remember and manipulate information with a general behavioral goal in mind, versus long-term memory is a more permanent form of storage for days, weeks, or even a lifetime. And again, I wish I could show all kinds of disorders and everything. You're kind of running short on time. If you want to see some videos that are associated with like amnesias and stuff, there's all kinds of cool videos and stuff associated with that. All right, so the process of converting immediate or working memory into long-term memory involves a process called consolidation. So formation and storage of declarative memory appears to require the hippocampus. Immediate and short-term memories are likely stored in this region. So formation and storage of non-declarative memory doesn't rely on the hippocampus, and memories seem to be stored somewhere in the cerebral cortex, cerebellum, or basal nuclei. Long-term potentiation is a mechanism by which the hippocampal neurons encode long-term declarative memories. This seems to involve an increase in synaptic activity between the associated neurons. Let's continue on. I think that's really it. The last one I wanted to touch was emotion. We kind of touched a little bit on emotion, and we touched the limbic system, but let's talk about this real quick. So emotion is a complex combination of three separate phenomena: visceral motor responses, somatic motor responses, and feelings. Okay. So visceral motor responses are something like your actual body is doing, like blushing, or your heart racing, or sweating, like your palms get sweaty. It's mediated by the hypothalamus. So somatic motor responses are smiling, laughing, frowning, crying, things like that. They're mediated by the hypothalamus and the limbic cortex through the reticular formation. Feelings are, again, highly subjective, kind of like pain, it's highly subjective. They're the most complex, and they're integrated with sensory and or cognitive stimuli. So feeling sad, remembering a lost pet, or feeling tense and watching a suspenseful movie is an example. And the amygdala receives input from the brainstem, thalamus, cerebral cortex, and basal nuclei, and analyzes the emotional significance of that stimuli. It creates associations between different stimuli and projects those to the prefrontal cortex. Again, you could take a whole class on all of that, but we only have a few minutes.